Assalamualaikum. My name is Nur Afifah binti Abdul Patah, and my title for final year project is Study of Lipid Production by Various Type of Microalgae. This is the outline for today's presentation. First, I will share about the introduction. Second is the problem statement. Third is the objective and scope. Fourth is the method. Fifth is result and discussion. And lastly, conclusion. As an introduction, there have three generations of biofuel. For the first generation biofuel, it is known as conventional biofuel, which is made up of sugar, starch, or vegetable oil. First generation biofuel are produced through well-understood technologies and processes like fermentation, distillation, and transesterification. These processes have been used for hundreds of years in many uses, such as making alcohol. Sugar and starch are fermented to produce primarily ethanol and in smaller quantities butanol and propanol. Ethanol has one-third of the energy density of gasoline, but is currently used in many countries, including the United States, as an additive to gasoline. A benefit of ethanol is that it burns cleaner than gasoline and therefore produces less carbon dioxide gas. Another first generation of biofuel, called biodiesel, is produced when plant oil or animal goes through a process called transesterification. Next is the second generation of biofuel, where they include woods, organic waste, food waste, and specific biomass crops. Fast-growing trees such as poplar trees need to undergo a pre-treatment step, which is a series of chemical reactions that break down lignin that hold plants together in order to make fuel. The pre-treatment step involves thermochemical or biochemical reaction that unlock the sugar embedded in fiber of the plant. After this step, the process to generate plant ethanol resembles that of first-generation ethanol production. The third generation biofuel is specially engineered crops such as algae as the energy source. These algae are grown and harvested to extract oil within them. The oil can then be converted into biodiesel through a similar process as first-generation biofuel, or it can be refined into other fuel as replacement to petroleum-based fuel. Disadvantages of first and second generation of biomass includes pose a threat to food price since the biomass used are food crops such as corn and sugar beet. They also have the potential to have a negative impact on biodiversity and competition for water in some regions. Biomass for first and second generation biofuel require lots of land to grow, and this provide only limited greenhouse gases reduction. And lastly, first and second generation biofuel are also a more expensive option than gasoline, making it economically unfavorable. Advantages of third generation of biomass includes ability to grow throughout the year, therefore algal oil productivity is higher in comparison to the conventional oilseed crops. Next is higher tolerance to high carbon dioxide content. Third is the consumption rate of water is very less in algae cultivation. And last is different source of wastewater containing nutrients like nitrogen and phosphorus can be utilized for algae cultivation apart from providing any additional nutrient. Next is photosynthesis process in microalgae. In the process of photosynthesis, non-polar lipids such as TAG stored in the cell. Yet this product has high commercial value as TAG can be transferred into fatty acid metal ester, which is a fundamental component in biodiesel. Esterification was the process used to convert TAG to FAME. Efficient cultivation in large-scale produce higher lipid amount to supply the biofuel globally. Microalgae produce two groups of lipid, which is polar and non-polar lipids. Glycerophospholipids is the example of polar lipid, which is important in the cell structure, and triglycerols is the example of non-polar lipid that store the energy of the microalgae. Photosynthesis reaction in microalgae contribute in generating the biochemistry. For the problem statement, the global human population is expected to increase more than 9.8 billion in 2050 with the current growth rate. The increment in the population requires more energy and more toxic gases emission. One of the main sources of this problem that may occur is usage of fossil fuel as the energy source. Carbon dioxide is the major component to cause the global warming and can be used as an indicator to check the level of greenhouse gases level. The way to cater the problem is by having a new source of energy that emits emit less GHG and toxic gas emission, and can be renewable. Biofuel is the alternative that had drawn the research attention over past years. Many of the regions in the world have started to use the biofuel as the alternative energy source, such as Europe and South Asia that use palm oil, Brazil that use sugarcane as the source of energy, and as for Japan, China, and United States, microalgae has been used. Biofuel that is produced in the world use the different organic matter. They have two objectives for this study. First is to study the lipid production and various freshwater and marine microalgae species. And second is to increase the production of lipid by optimization of the growth parameter, which includes salinity and iron content. Next is the methodology. First, both freshwater and marine microalgae are cultured in BG6 medium. Microalgae in this culture are considered as a constant. Then the culture are grown at the room temperature and 12-hour photo period. They are also being supplied by continuous aeration. Next, the concentration of iron chloride hexahydrate in AF6 medium are manipulated to determine the iron factor. Three different concentrations of iron chloride hexahydrate is used, which is 5, 10, and 15 mg per liter. For the salinity concentration, sodium chloride is added into the AF6 medium with 0.06, 0.08, and 1 mole. Lastly, to determine the growth of the microalgae, it is monitored every 2 days at 740 nanometer optical density using UV spectrometer. For the lipid production, the sample is taken every 7 days by harvesting the algae after centrifuge at 12,000 for 5 minutes before continued with the lipid extraction process. Next is result and discussion. So for the growth rate of Chlamydomonas mexicans and Synelesmus obliquus, both algae species will improve as the concentration of NaCl increased from 0 to 25 millimole. The growth rate of C. mexicana was relatively higher when compared with S. obliquus, and the salt stress ranging from 0.43 and 50 millimole NaCl. No addition of NaCl to BBM results in an adverse impact on the growth rate of microalgae. When NaCl omits from the medium, the growth rate of S. obliquus was inhibited compared to the control and other experimental conditions after 12 days of cultivation. After 20 days of cultivation, the dry weight of C. mexicana and S. obliquus was much higher for BBM amount with 25 millimole NaCl than observed for BBM without NaCl. Next is the salinity impact on lipid content and fatty acid composition of microalgae. So fatty acid profile and lipid content has been used as potential indicator of biodiesel productivity. The results shown revealed that the productivity of lipid from microalgae was dependent upon the concentration of NaCl in culture media, which is constant with previous report work. Lipid production from microalgae was improved by the addition of NaCl up to 25 millimole. The lipid production as a function of NaCl concentration was directly proportional to the algae biomass concentration, ranging from 0.06 to 0.8 gram per liter. The highest production of lipid was achieved by C. mexicana at 25 millimole and NaCl. A similar trend was also observed for S. obliquus, showing a lipid content of 34% at the same salinity. Next is the result and discussion for Nanochloropsis, which is a marine microalgae. From the graph, it was observed that the algae cells exposed to the hypersaline media experienced a higher growth rate in comparison to the algae cells that were exposed to the hyposaline growth media, as well as experienced a higher growth rate in comparison to algae cells that were grown in the control media. Calculation of the average rate of change yield a growth rate of 502,162 cells per day in respect to algae cells grown in hyposaline media, 1 million cells per year in respect to algae cells grown in the control media, and 2 million cells per year in respect to algae cells grown in hyposaline media. Quantitatively, this led to the observation that the number of algae cells grown in hypersaline media approximately double in respect to the algae cells grown in the culture media and approximately quadruplet with respect to the algae cell grown in the hyposaline media. Hence, as a conclusion, both microalgae are the microalgae that has the potential to be used as a feedstock to produce the biofuel as an alternative to fossil fuel. Unicellular photosynthetic microorganisms such as microalgae are proven to yield high amount of lipid to be used as a sustainable fuel. There are many ways that can be used to increase the production of lipid in microalgae. As been discussed before, the nitrogen, iron and salinity are some of the factors that can be manipulated to increase the lipid production. Each of the microalgae has a different rate in producing the lipid, hence more microalgae need to be studied to find the best microalgae that can produce a lot of lipid. Acutodesmal albicus is one of the freshwater microalgae that has a good potential to be further studied. This microalgae can be found in Malaysia and has high lipid content in the natural condition. Aside from that, Nanochloropsis is the marine microalgae that has high lipid content with a range of 39.4% to 44.9% of dry weight biomass. That's all from me. Thank you.